Having understood the chemical and functional nature of enzymes, we will now move on to the kinetics of enzyme catalyzed reactions. The enzyme kinetics deals with a quantitative analysis of the effect of various parameters on the rate of enzyme catalyzed reactions. It essentially involves molecular dynamics between the various species involved in the reaction. One of the principal species, as you know, is enzyme itself, and the interaction of enzyme with all other chemical species, that is the substrate, the product, if there is an inhibitor, all those molecular species, the dynamics of these interspecies interrelationship or interactions is involved in the study of enzyme kinetics. One of the essential feature for study of enzyme kinetics is the experimental data. To start with any kinetic analysis, one needs to initiate with some basic experimental observations, and to take some experimental observations, the key parameters that are usually involved in the study are the enzyme concentration, substrate concentration, product concentration, concentration of inhibitor or activator if there is one, whereas enzyme, substrate and product are the universal chemical species involved in the case of an enzyme catalyzed reactions. Then you have a set of environmental parameters like hydrogen ion concentration reflected in pH or the temperature of reaction. Then in most of the enzyme catalyzed reactions, we assume the water concentration to be very large, almost in excess, and for most of the analytical purposes it is ignored. But at least in two distinct cases where we are handling very high substrate concentrations of the order of say greater than 50 or 60% weight by volume, or in cases where we are carrying out reactions in the non-polar medium, the water activity also becomes a key parameter for study under enzyme kinetics, because then it controls the rate of the chemical the enzymatic reaction, and therefore it must be taken into account. So basically what I am trying to mean that when we talk of enzyme kinetics, we are looking into the molecular dynamics of various species that are involved in the reaction, their interconvergence and the effect of those interconversions on the rate of the reaction as a function of different parameters that are listed here. Now, there are three major constituents in any kinetic analysis. As I mentioned earlier, the first and probably the most important are the experimental data. With the start of the experimental data, one assumes certain mechanistic hypothesis, that means how various molecules involved in the reaction are interacting, and you propose some kind of a mechanistic hypothesis, or in other words you can say in a simplistic term the sequence of reaction steps that are involved in converting substrate into product, and based on this mechanistic hypothesis or reaction steps we arrive at using mathematical analysis a rate equation. Now, this rate equation again has to be verified with more experimental data. If it befits the experimental data, if it predicts or gives you a close relationship with the experimental data, it is acceptable. Both the rate equation as well as the mechanistic hypothesis we have assumed are acceptable. If not, we go again back to the mechanistic hypothesis, modify it, revise it in the light of the deviations, and then again go back to a new rate equation, and therefore such a analytical sequence continues till we are able to match our rate expression with the experimentally observed data. Now, in a very simplistic term, essentially the rate equation is a mathematical expression that depicts the reaction process in terms of rate constants. I think we must appreciate that the rate equation has a new dimension in terms of rate constants. A large number of rate constants might be involved depending on the number of reaction sequence steps, and of course substrate and product and other chemical species if involved. it also serves as a link between experimentally observed data and the plausible reaction steps, sequence of reaction steps. So, these three parameters or these three steps are the critical steps in study of any kinetic system, and of course, applies to also the enzyme kinetics. Now, I think the second question that might emerge in your mind, why do we study the enzyme kinetics? What is the function? I think the first and foremost and probably the most trivial function could be that could be understood is to get certain optimum set of parameters for carrying out the reaction. For example, when we consider what is the optimum temperature of the reaction, what is the optimum pH for the reaction, that is probably one of the primary objectives of establishing the whole kinetic analysis. Then of course, as I mentioned that you always assume a mechanistic hypothesis to arrive at a rate expression. It also gives a number of inferences about the reaction mechanisms, and as you will see notice further the current accepted rate expressions for enzyme catalyzed reactions involved a kind of a hypothesis where enzyme substrate complex has to be formed. Now, obviously, it gives you a very big inference when we look at the reaction mechanism also, and that reaction that has become a very sort of important part of the study of reaction mechanism itself. It can also give you a lot of sort of insight into the biological phenomena, the kind of reactions that these enzymes carry out inside the cell at a molecular level, their intercellular concentrations, their physiology, directions and regulation. A lot of issues about the function of these enzymes within the cell can be understood and inferred from the kinetic analysis of the enzyme. Then of course, from the point of view of biochemical engineers, a probably very important requirement for enzyme kinetic study is to design enzyme reactors. The design of enzyme reactors, just like even in the case of chemical reactors, is based on kinetic pattern. The kind of kinetics the reaction follows dictates very often the choice of reactor types, the design parameters for the reactor, and they all involve the enzyme kinetics, and that becomes quite an important tool for reactor design. Then finally, from an academic point of view, I think enzyme kinetics has also played a very important role in classification of the enzymes, particularly when the same enzyme is obtained from different sources. Their kinetic characterization makes you gives you a very good tool to distinguish between them and use and so that you can understand and identify the enzyme based on the kinetic characteristics. Also, even in as an analytical tool you can use, when you will see that when we want to determine many of the metabolite concentrations using enzymes, the basis for the design of the experiment is the kinetic parameters. We will probably talk when we will come to the actual kinetic aspects. Now, the third point which one must look at before studying the actual enzyme kinetics is some of the historical perspectives that have gone into the study of enzyme kinetics. Well, the understanding in the mind of the people, scientists, that there are some biological catalysts which catalyze a number of reactions, particularly the key reaction which people have understood then, thanks to the availability of alcoholic beverages, that the yeast possesses some enzyme which hydrolyzes sucrose into glucose, and of course, in those days the enzyme was understood, the alcoholic fermentation enzyme was called as zymase, although that nomenclature or that kind of understanding is no longer valid, but the first enzyme in the whole reaction sequence was invertase, which hydrolyzes sucrose into glucose and fructose, and that was essentially the a key enzyme which people were ready to study. And many of the kinetic analysis in the early stages, and when I say early stages I am referring to the late 18th century, early 19th century, when the study became, people tried to understand the kinetic behavior. And I think one of the earliest recorded interpretations or results of the kinetic analysis of enzyme was by O'Sullivan and Thomson. O'Sullivan and Thomson as early as in 1890. Now, he also studied the same enzyme and probably concluded in a very broad fashion that the enzyme reaction rates are influenced by acidity and enzyme concentration. These were the two parameters which today we understand pH and also substrate concentration, and of course, the temperature also is understood, and of course, he did not propose any basic hypothesis. What he suggested was based on his experimental observations that with every 10 degree rise in temperature, 10 degree centigrade rise in temperature, the reaction rate doubles, which is still, as a rule of thumb, it is still valid today. Then also he also gave a good clue which became a very landmark in the applications of enzyme, was that enzymes are more stable in the presence of their substrate. In fact, many of the enzyme preparations then could be stored in the presence of substrate, and that was another conclusion or the observation he gave way back in 1890.
So, how will we store an enzyme in presence of the substrate?
We store enzyme in the presence of substrate at low temperatures, so that the reaction rate is stopped for storage purpose. The low temperature was always a understood phenomena that at low temperatures the enzymes will have a much better stability, but the addition of substrate made them some way. Of course, they did not have any concept of enzyme substrate complex in those time, and he only gave a hypothetical suggestion that one could store at low temperature the enzyme in the presence of substrate, and of course, his observations were all based on hydrolysis of sucrose by invertase. Then the second major kinetic observations came from Brown, also in the early 19th century, and he proposed essentially for the first time the concept of an enzyme substrate complex, and he also proposed, based on experimental observations, that the reaction rate for an enzyme catalyzed reaction, that is conversion of a substrate into product, essentially follows a saturation kinetics. That means, the rate increases initially and reaches to a saturation level. Of course, he did not give any experimental any hypothesis or any sequence of steps, but he was able to understand that the basic reaction kinetics follows a saturation behavior. The rate increases to a maximum level and then stops at that, and beyond that there will be no increase in the reaction rate. And of course, at low substrate concentrations, basically the enzyme reaction rate is proportional to substrate concentration. That is a phenomena still we understand now with our detailed understanding of the enzyme kinetic even today. Now, they were the broad observations by Brown. Then of course, the major breakthrough in the enzyme kinetics was Michaelis and Manton. As early as 1913, you imagine that at that time we had great limitation of analytical methods. In most cases, there were not very strong analytical procedures available to quantitatively measure the concentration of product. The concept of buffers was in a very primitive stage. Buffers were not a very common tool to be used in the kinetic analysis. Then they did not have much understanding of the stability of the enzyme as well, and all those limitations led to the and of course, thirdly, that the enzymes they used were really not available in the highly purified state. What they were talking all with the crude enzyme preparations, and so the observations they could get were very limited. And now, what Michaelis-Menton's major contribution was, one is that he confirmed that Brown's observation that enzyme substrate interaction undergoes to formation of an enzyme substrate complex, that is E plus S goes to ES complex, and he also proposed that this interaction is reversible in nature, and basically he was the first person to propose a hypothesis, mechanistic hypothesis, which is followed by enzyme catalyzed reactions, and then this enzyme substrate complex then breaks irreversibly to form the product and enzyme is released back. That was the hypothesis he proposed and probably is one of the landmark contributions in the study of enzyme kinetics even today. And of course, secondly, he could also generate data using buffered substrate, so that he for the first time carried out reactions under controlled pH. Then the third thing what he introduced another concept. So, as you notice that the first contribution was enzyme substrate complex, the second was buffered substrate, and the third and another important feature was that he introduced when we talk of rate of reaction, he mentioned that we must talk only of initial rate of reaction V0, which is your dP over dt at t is equal to 0, initial reaction rate. He says that once a significant quantity of product is formed, the kinetic behavior changes. The enzymes follow a kinetic behavior which can only be understood at the initial reaction rate level. That means, when you carry out the reaction, the rate of product formation at time 0, and that was another thing which he. And then the fourth thing was, as I mentioned right in the beginning, that he confirmed the concept of enzyme substrate complex. He also mentioned that the interaction between enzyme and substrate to form enzyme substrate complex is in perfect equilibrium. It reaches equilibrium at a very short time, and then the reaction proceeds further to form the product. And based on this hypothesis, he developed his rate expression. Basically, if you can write down the rate constants here, K1, K2, K1 minus 1 and K2 for the 3 reaction steps that are involved. In the case, one can write down the rate expression using the equilibrium hypothesis. That means, at equilibrium, your K1 into E into S is equal to K minus 1 into ES, and this by series of interpretations and using enzyme conservation equation, E0 will be equal to E plus ES. I mean, the total enzyme concentration then at equilibrium is distributed between enzyme substrate complex and the free enzyme, and substituting the value of ES from your equilibrium analysis, you can come down to a initial reaction rate V0 will be equal to K2 into concentration of ES. If you look at your previous the original mechanistic hypothesis proposed by Michaelis and Menton, K2 P plus E. Sorry. Now, so you can write K2 ES will be the rate of initial rate of reaction, on which substituting the various parameters you get K2 E naught S naught upon K minus 1 upon K1 plus S naught, sorry, plus S naught. So, you get the initial rate expression where the initial reaction rate in terms of initial enzyme concentration and substrate concentration are given. Now, the rate constant here, as you notice, is K1, K minus 1 and K2. The term K2E0 was the saturation reaction rate, the saturated reaction rate when all the enzyme is saturated with the substrate that is bound to substrate, a hypothetical situation which will never emerge, because there will be always a equilibrium between enzyme, free enzyme, substrate and the enzyme substrate complex. But hypothetical situation is that if all the enzyme is say saturated with the substrate, then you have a maximum reaction rate which is called Vm, and also K minus 1 upon K2, the ratio of the two rate constants K2 and K minus 1. That means the ratios of two rate constant which is involved in the breakdown of the ES complex either ES complex can go to product formation or it can go to back to the substrate complex. Now, the ratio of the two was considered as Kd, the dissociation constant, essentially the dissociation constant of ES, and that minded that he never proposed at the time of Michaelis-Menten, there was no proposal of Michaelis-Menten constant. He only proposed that this is a dissociation constant of enzyme substrate complex, and the kind of profile, a graphical representation of such a reaction will be substrate concentration versus initial reaction rate. It will follow a saturation behavior where the maximum reaction, the saturation behavior, the saturated reaction rate reaches to a value of Vm, which is essentially nothing else but K2E0, and the reaction rate at half the Vm, half the value of the maximum reaction rate was defined as Kd, dissociation constant. So, basically with that concept he was able to determine the dissociation constant of your enzyme substrate complex formation, and he also proposed that now this substrate concentration at which the reaction rate reaches half the Vm value, essentially which is the dissociation constant, it also has a direct implication with the affinity of enzyme to the substrate, because after all it is the rate, it is the substrate concentration at which the reaction reaches to half of the saturation behavior, and therefore it directly implies the affinity behavior and being represented in terms of dissociation constant. Now, of course, this was a major contribution which for a long time was valid until the time another major contribution which came in the enzyme kinetics and also is by Briggs and Haldane. In 1925, mind it that Haldane, J.V.S. Haldane was one of the scientists, British scientist of course, who did all of his work in India, and they proposed a hypothesis that yes, the Michaelis-Menten hypothesis is very correct in most of the respect, except that they did not agree that the interaction between enzyme and substrate is that of equilibrium behavior. Instead of equilibrium behavior, they mentioned in the case of a rate terms a steady state behavior, and that means again they assume the same thing, E plus S going to enzyme substrate, I will just write as X, and goes to E plus P. Now, they also expanded the whole the hypothesis. Instead of the enzyme substrate breaking down through a irreversible step, in many reactions which are known to be reversible in behavior, as many times I have earlier also stated, the isomerization of glucose to fructose is a true reversible enzyme reaction, and there are many such examples where the reaction can be catalyzed in either direction, and therefore he also considered that even the second step in the sequence is also reversible, and we need not limit it to a irreversible behavior. And also he mentioned that instead of having a equilibrium behavior here, we have a steady state behavior, which essentially means the rate of change of x is 0 with time. That means the concentrate that the after a very short interval of time when the reaction starts, the concentration of enzyme and the enzyme substrate complex reaches a constant value. And if you look at the kind of a scenario he proposed, that if this is the time scale for the enzyme catalyzed reaction and this is a general concentration axis, then enzyme concentration goes down, the enzyme substrate concentration builds up, and this is enzyme concentration, this is an ES concentration. So, essentially after, and this is that short time what I am referring. Now, mind it that when I am showing it here in expanded view, that this time scale is probably only a few seconds. This time scale up to here is only a few seconds, which we today and consider as, he named it as a induction period. Of course, you can call it a transient or pre-steady state period also, but as soon as this transient period, which is a very short time, passes, both the enzyme substrate complex rate of formation of enzyme substrate complex comes at a steady state, and this is becomes 0, and based on that he wrote his rate expressions, and of course arrived at almost identical rate expression as Michaelis and Menten, with slight difference which we will notice, and you can write then at with the assumption of dx over dt is equal to 0, you can again write and also with E0 is equal to E plus x is enzyme substrate complex, and then also you can also take the conservation equation for substrate, which will be equal to the substrate concentration plus product concentration, and you can, if you just write all the expression in a rate term, you will find the concentration of x will be equal to K1, and the rate constant here are K1, K minus 1, K2, K minus 2. So, it will be K1 E naught S plus K minus 2 E naught into P upon K minus 1 plus K2 plus K1 S plus K minus 2 P.
So,
yes.
So S naught should be S plus P plus X.
Yes. Right. But X, as a matter of fact, one can always neglect because it is too small a concentration compared to S. Because also we assume, I think in most of these analysis, that your S naught is much greater than enzyme concentration. The concentration of substrate, being a small molecule usually, is much larger compared to the enzyme concentration, which is used as a catalyst in very small concentrations, and usually in any enzyme catalyzed reaction the concentration of the enzyme will be of the order of a very few millimolar or even less than that. So, you get the concentration of enzyme substrate complex, and based on this hypothesis you get the initial reaction rate. He also followed the initial reaction rate concept as K2 E0 into S upon K minus 1 plus K2 upon K1 plus S. Now, this is after, if you take a general reversible equation and put P is equal to 0 at P is equal to 0, because if you are considering the forward reaction only, where you start with the substrate at the initial rate, the product concentration is 0, and if you in your general expression, if you put P is equal to 0, you will arrive at the initial reaction rate. So, what you notice is the difference between Michaelis-Menten kinetics and that proposed by Briggs and Haldane lies in the fact that in the case of Michaelis kinetics, they proposed the diffusion constant K minus 1 upon K1, which was converted or which got transformed in the form of K minus 1 plus K2 upon K1. Now, this parameter essentially K minus 1 plus K2 upon K1, in honor of Michaelis-Menten to recognize their contribution, because bulk of the idea they got in their whole analysis from the Michaelis-Menten assumptions, they named this parameter as Michaelis-Menten constant. In fact, the very fact, a very great confusion that Michaelis-Menten himself never proposed or gave that name as Michaelis-Menten. He always talked about diffusion constant, which is a very fundamental parameter in the physical chemistry. But it was only Briggs and Haldane, they proposed that this and the constant which they proposed was not developed by Michaelis-Menten. This was Briggs and Haldane's parameter, a lumped parameter of rate constants K minus 1 plus K2 upon K1 was named as Michaelis-Menten constant, which of course is analogous to the diffusion constant, and the only difference is, what is the difference between the two? The difference lies in the K2 upon K1. K2 upon K1, the difference between the two parameters is K2 upon K1, and essentially the two parameters will become identical if we assume that the K2 is much less than K minus 1. If the K2 is assumed to be very small rate constant compared to k minus 1.
So, but is that true?
In most cases it is true incidentally, and that is why even the diffusion constant concept is feasible, because in most cases the rate of breakdown of enzyme substrate complex, and that is the reason why many of the enzyme reactions are noted to be very slow in terms of the rate processes. They are noted to slow. The term K2, the rate constant K2 is usually in many cases it is fast also, of course, incidentally, like for example in the case of inverters itself K2 has a significant magnitude, but in many enzyme catalyzed reactions the K2 is very small compared to k minus 1, and therefore the two hypotheses provide you a very I mean similar sort of inference as far as the function of or the kinetic function of enzymes are concerned. The concept of Vm, the maximum reaction velocity, the concept of the Michaelis-Menten constant, the physical significance of these two parameters, which are the critical parameters, also remain the same even with the Briggs and Haldane. So, I mean just to summarize the results of the Michaelis-Menten kinetics and Briggs and Haldane kinetics, we have two kinetic parameters which can characterize any enzyme catalyzed reaction. One is the maximum reaction velocity, which is K2 E naught, other is Km, which is k minus 1 plus K2 upon K1. Now, another point I like to probably make it clear here, Vm is not a kinetic constant. Vm is a parameter, is not a kinetic constant, it is a function of enzyme concentration. Is a function of enzyme concentration. If you increase enzyme concentration, the value of Vm increase. The kinetic parameter here is K2, the rate constant which is responsible for breakdown of enzyme substrate complex into the product, and very often the K2 is also called as turnover number, which is the rate limiting step in the enzyme catalyzed reaction, and the composite term K2E0 characterizes an enzyme in terms of saturation, but again that Vm value is at a particular enzyme concentration. When you mention a Vm as a characteristic, it refers to a particular enzyme concentration. Km is a true kinetic parameter, which is a lumped parameter of the three rate constants and is typically characteristic of any enzyme function, and very often that is why Km is used as a characterizing parameter for most of the enzymes that are available for. Now, this kind of a kinetics, if you look at, let us say V0 is equal to K2 E0 into S upon Km plus S, I think you will also notice a very significant trend. If you look at, again go back to its graphical representation, a saturation kinetics represented by a rectangular hyperbola. This is Km at Vm by 2. Now, unlike most of the chemical reactions, you will notice that this represents a case where the order of a reaction changes with substrate concentration. See, most of the chemically catalyzed reactions or chemical reactions represent a particular order of reaction, 0 order, first order or second order. In this case, you will notice that at low substrate concentration, when substrate concentration is much smaller, is very small compared to Km, of course you can keep Km as the sort of a datum. If it is very small compared to Km, you can arrive at V0 is equal to K2 E0 into S, a first order kinetics, simple first order kinetics, and because K2 E0 is a constant at a particular given enzyme concentration, K2 E0 is a constant and substrate concentration is a variable, and so it becomes a first order kinetics, and that is what is the portion represented by initial this area, first order kinetics. Similarly, on the other extreme, if you go, let us say, to this the concentration beyond this point, when substrate concentration is much greater than Km, your V0 becomes K2 E naught, approximately equal to Vm, which means essentially a zero order kinetics. And in between the two, the enzyme follows some kind of a mixed kinetics, and that is one very characteristic feature of enzyme catalyzed reactions, that instead of following a fixed kinetic order, it follows a changing order of reaction kinetics, and at the two extremes, and therefore now this has a very important feature. In most of the analysis, most of the subsequent analysis of the kinetic parameters or the reaction rates or the reactor design, we often to simplify the situation, we often either go to the zero order enzyme or go to the first order enzyme, very often, because this expression essentially is comparatively much more complex to analyze rather than a zero order or first order enzyme, and for most of the analysis to understand the system, either we simplify it to a zero order enzyme and experimental data can be collected either at a very low substrate concentration range or at a very high substrate concentration range. Now, when we see that the reaction rates and also this understanding can be used even for use of enzymes as an analytical tool or the assay of the enzyme itself. All these information can be used very significantly for those purpose. Now, just to explain further the or just to continue on the Haldenase Briggs and Haldenase kinetics based on the reversible enzyme catalyzed reaction, that means where if you recall what we got as V naught is equal to K1 K2 into S minus K minus 1 K minus 2 P whole E naught upon K1 into S, sorry, K1 into S plus K minus 2 into P plus K minus 1 plus K2. Now, if you consider the original the reaction rate expression which was developed by Briggs and Haldane for a reversible reaction, that means you can put the value of any either substrate concentration, you can carry out the reaction either from the in the reverse direction or in the forward direction, starting from S or starting from P, and you can arrive at a rate expression in terms of substrate and product concentration. Now,
this will not be V0.
Pardon? No, this will not be V0, you are right. This will be V general at any given time. Now, here you can put now you can, from this expression you can determine the equilibrium constant for the reaction. That means equilibrium constant is the one where the rate of forward reaction and the backward reaction are identical, and the equilibrium constant, the value of the concentration of product upon substrate at V is equal to 0 will be your P at equilibrium upon S at equilibrium, and that can be derived to arrive at K1 K2 upon K minus 1 K minus 2, or in terms of the basic kinetic parameters, maximum reaction velocity based on substrate into microelectrode maintenance constant based on product upon Ks into Vp. The expression can be arrived from the main rate expression, which will give you the equilibrium constant either in terms of the rate constant, and of course the equilibrium constant can be monitored experimentally, and that can also give you an idea of measuring or determining the individual kinetic parameters experimentally.
So, how did we arrive at this P by S equilibrium is equal to K1?
This will be, this you can arrive at, you can put V is equal to 0, V is equal to 0 and solve it and determine the value of P equilibrium. At equilibrium the reaction rate will be 0. and you can arrive, and I think I leave it to you to develop this. I don't think it will be a very difficult task to do that. Now,
What is Vs?
Vs is the maximum reaction velocity corresponding to substrate. That means, if you carry out the reaction with substrate, initially with the S substrate, the maximum reaction velocity it reaches is Vs, and similarly the Ks is the Maclaurin-Bennett constant corresponding to substrate S, and the PR corresponding to the P when you start the reaction with. You see, these data Vs, Ks, Kp and Vp will be determined experimentally on the basis of initial reaction rates and not as a general reaction. These parameters will be determined on the basis of initial reaction rates corresponding to either S or either P. You can write here, I think, VMS or VMP, just to be. Now, I think next step came that obviously one will like to understand the kinetics, the enzyme kinetics, as I mentioned, from on the basis of the experimental data. Now, how to make a link between the experimental data and the kinetic rate expression that was obtained by these two scientists separately. But of course, basically today Briggs and Halden's kinetics has become a universally accepted reaction kinetics, which is followed all over the world to understand the enzyme catalyzed reactions. Now, the major issue is how to use those data for primarily for determining the Km value, the value of the Km or the Vm value corresponding to a particular enzyme concentration. Now, essentially experimentally you cannot determine the Vm value because it is a saturation rate asymptotic to the axis, and therefore, you can never reach to that value experimentally. You may reach closer to it as you increase the substrate concentration, but you will never be able to approach in the terms of the. So, I think an attempt was made for the treatment of experimental data so as to test or verify the hypothesis that has been proposed by Maclaurin-Bennett initially and verified and approved or vetted by Briggs and Haldane. And I think so the simplest way to do that was that these non-linear equations, if they could be converted by some means, by some modification into a straight line relationship, I think the life will be much easier to analyze the experimental data, and the first attempt for that was given by Lineweaver and Burke. Pardon? Just I'll give this. I have a watch in front of me, and Lineweaver and Burke, they made a very simple approach that you convert, that is, your V naught is equal to K2 E naught into S upon Km plus S. Both the sides you take reciprocal, and you can just write 1 upon V net or not, 1 upon V naught equal to Km upon S into 1 upon K2 E naught plus 1 upon Vm, or 1 upon K2 E naught. So, therefore, now this represents a straight line relationship between 1 upon V naught and 1 upon S. That means, when you plot 1 upon V naught and 1 upon S, you get a straight line relationship, which essentially with a slope is equal to Km upon K2 E naught or Km upon Vm, and intercept here which refers to 1 upon Vm. And of course, on the x axis also this will be an intersect which will be minus 1 upon Km. So, in a very simplistic way, if you can take a double reciprocal plot, one can analyze the experimental data that are obtained on the rate of reaction as a function of substrate concentration and verify the hypothesis. Now, for most of the cases, such a hypothesis has been verified and found to be proven, of course, under certain limited conditions which have to be in line with the assumptions made by the scientist, because as we will see further that you will notice that while Michaelis-Menten kinetics is the most accepted kinetic pattern of the enzyme catalyzed reaction, most of the enzyme catalyzed reaction in practice deviate from the basic assumptions, and only under certain set of conditions they obey the enzyme kinetics proposed by Michaelis and Menten, Opdix and Haldane. And so, I think with that we will stop at this point.